Okay, let's be honest for a second. We've all had that thought, haven't we?
Oh, yeah.
Imagine getting all the incredible health benefits of exercise, the metabolism boost, the endurance, better energy levels,
Hmm.
but without actually having to, break a sweat, just maybe take a pill.
Right, the dream scenario.
For the longest time, that felt like pure fantasy, right? Something straight out of science fiction.
Absolutely.
But is science actually getting closer now? Could exercise and a pill be something we might eventually see?
Well, it's definitely moving from pure fantasy into, let's say, active research territory. Scientists are really trying to understand and target the body's own complex pathways that exercise switches on.
And today, that's exactly what we're doing. We're diving deep into one specific compound that's really making waves in this area. It's called SLU-PP-332.
Yep, SLU-PP-332.
And our dive today, it's based on some specific source material you shared with us, focusing on key excerpts from a document that really lays out the potential and also the current status of this compound.
Exactly. So our vision here is really to unpack all this information for you.
Yeah.
We want to explore what SLU-PP-332 actually is, what the initial research, especially in preclinical models like animals, has shown so far. And then, crucially, we need to ground that excitement and look at the very real limitations that exist right now.
Okay, sounds good. Let's start right at the beginning then. What is SLU-PP-332? The source calls it a synthetic compound. What's it designed to do in our bodies?
Right. So at its core, it's a molecule made in a lab. It's designed specifically to act as something called a pan-agonist.
Pan-agonist.
Yeah, meaning it activates multiple targets, and its targets are a particular set of receptors in your body known as estrogen-related receptors, or ERRs.
ERRs. Okay.
Think of them like tiny locks inside your cells. SLU-PP-332 is essentially a key designed to fit and open these specific ERR locks.
Okay, estrogen-related receptors, ERR. That name immediately makes me think, do they respond to estrogen?
Yeah, that's a really common and important question, and the source clarifies this point well. Despite that name, ERRs are generally not activated by estrogen itself.
Oh, interesting.
They're actually classified as what scientists call orphan receptors.
Orphan receptors, why?
Because when they were first discovered, they're natural activators. The body's own keys for them weren't known. So their activity is influenced by other internal signals, or, like in this case, by synthetic compounds, keys made in the lab, like SLU-PP-332.
Got it. Okay, so their orphan receptors don't typically respond to estrogen, but do respond to the synthetic compound SLU-PP-332. So what's the big deal about these ERRs? What kind of locks are they opening? What happens when you turn that key?
Well, they're a type of protein called nuclear transcription factors.
Oh, okay, bit technical.
It is, but basically it just means they hang out in the nucleus of your cells, the control center, and they directly control which genes get turned on or off.
Right, like gene switches.
Exactly. And the specific genes they control are absolutely critical for fundamental cellular processes, especially those related to how your cells make and manage energy. Things like mitochondrial metabolism and overall cellular energy production.
So activating these ERRs is like flipping these master switches that control cellular energy systems.
Yeah.
And SLU-PP-332 activates. Which specific ERs did the source mention? Was it 3 types?
That's right. It's designed to activate the 3 known ERR isoforms.
So
there's ER alpha, or ERI,
alpha.
ER beta, ERR, and ER gamma, ERO.
Okay, alpha, beta, gamma. Does it hit all 3 with the same force, so to speak?
Good question. And this is where the source gives us some important details about how it actually works. While it's called a pan-agonist, suggesting it hits all 3, it actually has a measurable preference. The document provides these numbers called EC-ER values.
EC-ER. Okay.
Now, without getting too bogged down in the chemistry, you can think of ECR as basically how much of the compound you need to get half of the maximum activation effect.
Right.
So the EC-EROs for ERA is around 98 nanomolar, or NM. For ERO, it's about 230 nm. For ERO, it's roughly 430 nm.
Okay, 98 versus 230 and 430. How do we read those numbers? Lower is better.
Lower means more potent. You need less of the compound to get that response. So yeah, 98 nanolam is significantly lower than 230 or 430.
I see.
This tells us that SLU-PP-332 is most potent at activating ERO. It shows a clear selectivity bias towards alpha compared to beta and gamma.
Okay. So it's like a master key that opens all 3 locks, but it works best. It's most efficient on the ERO lock.
Makes sense. And where in the body do we find ERO predominantly?
ERO is highly expressed, meaning it's found in abundance in tissues that have the highest energy demands, places like your skeletal muscle, your heart muscle, and also your kidneys.
Okay, muscle, heart, kidneys, energy-hungry tissues.
Precisely. And this is relevant because these are exactly the tissues where changes in energy metabolism have a really big impact on their overall function. Right. So SLU-PP-332 targets and most strongly activates ERI in these key tissues, and ERA controls genes related to energy production. So what does activating these pathways actually lead to? What are the downstream effects?
So when these ERR pathways get stimulated, it regulates the genes involved in processes that are absolutely fundamental for cellular energy.
Okay.
This includes things like fatty acid oxidation. That's how your body burns fat for fuel.
Right. Fat burning.
Oxidative phosphorylation, which is a key process for making ATP, your cell's energy currency, using oxygen.
The main energy molecule.
Yep. Also, mitochondrial biogenesis,
This is?
the creation of new mitochondria, the powerhouses of your cells.
Building more power plants.
Exactly. And overall energy homeostasis,
Yeah.
basically, keeping your body's energy budget balanced.
Wow. So by stimulating all their core energy processes, burning fat more effectively, making energy efficiently, building more cellular power plants, it's essentially sending a metabolic signal to the body that sounds remarkably similar to what happens after you've done some serious exercise.
That's precisely how the document describes it. It effectively mimics a post-exercise metabolic state by stimulating these critical energy pathways that are under the control of ERRs.
Okay, that's the mechanism then. Activating ERRs, especially ER, boosts these fundamental metabolic functions. Now let's get to the results. What has the research actually shown this compound can do in studies so far?
Okay, but here's the really crucial point we need to stress right up front, just as the source does. These findings are almost entirely from preclinical animal studies.
Right, not humans.
Mostly mice. Almost everything we know currently comes from work done in mice.
Understood. So keeping that big caveat in mind, what effects have researchers seen in these mouse models when they're given SLU-PP-332?
Well, the documented findings are actually quite compelling. Within that context, they've observed effects like increased energy expenditure.
Meaning the mice burn more calories.
Exactly. Enhanced mitochondrial respiration, which means their cells get better, more efficient at making energy, and also improved insulin sensitivity.
Ah, insulin sensitivity. That's a huge marker for metabolic health, something people strive for with diet and exercise.
Definitely. And then there are effects directly on muscle and physical performance, too.
Oh, yeah. Like what?
Studies showed it actually promoted the formation of what are called oxidative muscle fibers, specifically type CA fibers.
Type CAA. What does that mean in simple terms?
Well, muscle fibers come in different types, broadly speaking. Some are built for quick bursts of power, others more for endurance. Type CA fibers are sort of in between, but they lean towards being more oxidative, more fatigue resistant, the kind that are really key for sustained aerobic activity.
Okay, so it encourages the development of more endurance-type muscle fibers.
Sort of, yeah. And critically, in these animal models, treatment with SLU-PP-332 also enhanced aerobic endurance and their overall exercise capacity.
So the mice could literally run further or longer.
That's what the studies suggest, yes.
And I think the source mentioned something even more specific about a preprint study looking at the genetic level.
Yes, exactly. There's a 2023 preprint cited, published on bioRxiv, which is a platform for preliminary research. It found that just a single dose of SLU-PP-332 given to mice.
Just one dose.
One dose, yeah. Triggered a transcriptional response in their muscle tissue, basically. It activated a pattern of genes that was remarkably similar to the pattern of gene changes you see right after acute aerobic exercise.